Good morning. My name is Sylvia Anatara, and today I'm going to present my final year project. My title is Comparative Leaf Proteome of Irradiated Mutant and Wild-type Oryza sativa indica. My supervisor of my final year project is Professor Dr. Muhammad Hasnain. Oryza sativa, known as Asian rice, is one of the rice plant species that are widely grown in tropical climates. According to Mutaya and his colleagues in 2019, it is also one of the economical staple food for about 3.5 million people worldwide, as it contains primary sources such as vitamin, nutrition, and mineral. Asian countries have the highest production of rice because it supplies dietary calories as much as 50% and considerable importance in protein intake for more than 520 million people living in poverty in Asia. Rice has become primary source of income and employment. The, the objective of this study is to identify the gene that related to the growth of rice and to identify the production of rice starch. The literature review in this study is divided into three parts, which are Oryza sativa indica, benefit of rice, and the use of irradiated mutant of Oryza sativa indica. Rice growth is divided into four stages, which are germination, vegetative phase, reproductive period, and ripening phase. Rice seed germination occurs when roots and shoots grow from the seed. It depends on the location, such as the root will begin to develop when the seed is planted in non-flooded soil, while the shoot will emerge when the seed is planted in flooded soil. Next, vegetative stage refers to the growth of tiller, leaf, and plant height that take 58 to 85 days. The reproductive stage begins when the leaf exposes the developing panicles, which is known as the budding stage, followed by the heading stage, which refers to panicles that have fully emerged from the leaf stem and become visible. The pollination occurs approximately 7th day. The final stage is the ripening period, which is the grain is fully ripened and ready to be harvested, which normally take 15 to 40 days, depend on the humidity and temperature. Rice has many essential food sources: traditional medicine, Western medicine, and cosmetics. In food sources, rice flour is gluten-free and typically used to make noodles. Aside from that, rice cultivars are used as a thickening agent in sauces and pudding. Other than that, rice also has snack foods in Eastern Asia, such as rice cracker and rice cake. Rice can be used to treat skin conditions in traditional medicine. In contrast, laboratory studies in Western medicine have revealed that rice products may have anti-cancer properties and the ability to treat other conditions such as diabetes, kidney stone, and heart disease. Finally, rice extract can be used in cosmetics. Starch and oil, for example, are used in a variety of cosmetic and hygiene products. My third literature review is the use of irradiated mutant of Oryza sativa indica. A study by Sikora, Charade, Larson, Olson and Olson in 2011, Cheong, Ding, and Lee in 2015, plant breeding started in ancient times, parallel to human culture, as one of the earliest agricultural activities. Science applications on production of rice are done to improve its ability to grow on different types of stress conditions, as well as to increase the nutritional value in rice. As a result, lots of rice varieties such as Oryza sativa indica and Oryza sativa japonica have been developed to meet the consumer demand. Rice research has widely used in biotechnology applications such as proteomics to study in depth about its protein coding genes, as well as protein and gene expression. As stated by Chakraborty in 2015, the rice proteomics have changed which may be result from exposure eh, to different stress such as climate change, the usage of fertilizer, presence of pests, humidity and temperature in recent years. The material and methodology of this study is plant material, extraction of protein and quantification, process electrophoresis of polyacrylamide gel in two-dimensional, and lastly, comparison of protein. Plant leaf of irradiated mutant and wild type will be manually cleaned using distilled water, followed by sterilized using 70% ethanol. Both plant types will be instantly frozen in liquid nitrogen and stored at -80 degrees Celsius for subsequent analysis. These irradiated mutant and wild-type of rice were already available in our proteome laboratory at University Malaysia Sarawak, Malaysia. The extraction of protein and quantification is to purify a protein in order to identify the gene and to determine total protein concentration by using a standard Branford protein assay. Next, the process electrophoresis of polyacrylamide gel in 2D is to separate and display gene protein based on isoelectric point and size into resolved protein by SDS-PAGE. Last but not least, comparison of protein is to identify the protein spot on on polyacrylamide gel and to compare the structure of the protein. The expected outcome for my research is the irradiated mutant of the rice leaf had the most significant protein spot compared to the wild type of rice leaf. It will affect rice growth, which means the irradiated mutant grow faster than the wild type. Irradiated mutant of Oryza sativa indica can increase the yield of the rice. As expected from this study, it will help increase it will help increase and improve rice production growth and the nutritional content of the rice, such as protein and starch. Other than that, it will also help to overcome worldwide food scarcity due to the increasing human population and reduce malnutrition in the developing country. Last but not least, the rising production of rice will eventually open more job opportunity and financially improve the Malaysian economy. This is my work schedule for this research, and this is the list of my references. Thank you for listening to my presentation.